A goal with the recent PANC-DB update was to have process single-cell RNA-seq data available to researchers. This saves researchers a lot of time because the processing of the single-cell RNA-seq analysis requires a lot of training and time to perform. The processing of the single-cell RNA-seq analysis was performed by our colleague Abhijit Patel. The workflow that he followed to do this processing is available underneath Workflow and Protocol, and then single-cell RNA-seq analysis. His workflow includes how he concatenated the experiments together, how the low-quality cells were removed, how the gene expression was normalized, and how the cells were annotated. The output of his workflow is available under Data and then Interactive Analysis. On the Interactive Analysis page, we have the process single-cell RNA-seq data available as an R object, a Python-compatible format, and then also we have a link to in-browser analysis of the data. If you don't know what R and Python are, R and Python are popular coding languages used in bioinformatics. Now, the workflow that Abhijit created is done in R, and it outputs an R object. So I wrote a script which converts that R object into a Python object, and that Python object is then compatible with the CellXGene application. I'm going to open that now. So the CellXGene application is an application written by the Chan Zuckerberg Initiative team, and it allows us to view and browse single-cell data in the browser. Now, the way to read this is that in the center we have a UMAP plot. And so for each of these dots on the UMAP plot represents one of the 222,000 cells in this data set. And the clusters here means that these sets of cells are related to each other in terms of gene expression. But just because the cells are close to each other on this plot does not mean that they're closely related. For example, if cell A is here, cell B is here, and cell C is here, just because they're about the same distance from each other does not mean that A is as different to B as B is as to C. On the left, we have the cell annotations, and on the right, we have the gene annotations. So on the left, I want to go through each of the different cell annotations we have. We have age, we have assay that was used, we have cell type, we have disease state, we have donor ID, we have race, and we have sex. Now, when we highlight using this color icon here, we're able to color the plot by that cell annotation. For example, when I'm coloring the disease state, I'm coloring it by antibody positive in purple, control in red, green is type 1 diabetes, and in blue is type 2 diabetes. And this number here is the number of cells that are in each of these groups. And when we have one cell annotation highlighted, we're able to see the proportion of that cell in the other cell types. For example, we can see that for beta cells, the proportion of beta cells that are type 1 diabetes is very low. But the proportion of beta cells that are a sinner are slightly higher. Now, one thing, so I'm going to color by cell type, and remembering each of these colors for the cell types can be quite tedious. So one thing I'm able to do is I'm able to click this this bullet drop down here, and now each of the different clusters are labeled. So we can see our center cells, epithelial cells, leukocytes, endothelial cells, etc. Okay, now I'm going to show the gene navigation bar. So I'm going to search up CPA1. CPA1 is a gene which is unique to our center cells in the pancreas. I may refer to genes that are distinct to a certain cell type as marker genes. So what I'm going to do now is I'm going to click this droplet icon, and now the plot is colored by the gene expression of CPA1. And we can see that in this top right hand corner we have slightly higher CPA1 expression. I'm going to see more details about this. So I'm going to click this icon here, and a histogram appears. The way to read this histogram is that on the x-axis is the gene expression, and on the y-axis is the number of cells with that gene expression. So that we can see that we have many cells that have 0 gene expression of CPA1, and then we have some other cells that have a higher gene expression of CPA1. Now, if I want to just highlight the cells that have non-zero CPA1 expression, and we'll click and drag on the histogram here, and we can see that the top right hand corner of the UMAP is highlighted now. And I'm able to confirm that these are center cells by going over here and seeing the small histogram here, or when I highlight over our center cells, we can see that the top right hand corner is what gets enlarged. Now let's say I want to see this this small part of the histogram in more detail, and remove out the 0 counts here. The way that I'm able to do this is by clicking this bar plot icon here and clipping the continuous values from 85 to 99 and pressing clip. And now we can see more details about the CPA1 expression in this data. So we can see that we have CPA1 expression over here, but we have more gene expression of CPA1 over here. Okay, so next, let's say that we want to find which genes are distinct to healthy or center cells. The way that we're going to do this is by running something called differential expression. Differential expression uses statistical analysis to define which genes are distinct of different cell groups, or different groups of cells. So the way that we're going to do this is that I'm going to first select just control. I'm going to color by disease state here and take off the labels. So I'm going to select control and then our center cells, and then have this as the first set of cells, and then I'm going to have everything but their center cells as a second set of cells. I'm going to press this Venn diagram button here, and it's going to run. Now it's going to take some time because we are comparing the gene transcripts of 25,000 cells versus the gene transcripts of 81,000 cells. We did buy a new server just for the CellXGene application to give researchers plenty of power. I'm also going to right now change the continuous values back to 0 and 100 so it doesn't get confusing for us later. Okay. Now I'm going to confirm the results of differential expression. We can see that for the first population, which is our healthier center cells, we have PSSR2, Reg1A, and PSSR1. And we also have CPA1, which we know from before is a marker gene of our center cells. So let's say that we want to compare the expression of two genes. The way that I'm able to do this is that I'm going to have PSSR2 as the first set and CPA1 as the second set. So X and then Y. So on the x-axis we have so each of these points, again, each of the 220,000 points represents one cell. And on the x-axis is from the lowest to the highest expression of PSSR2. And on the y-axis we have the lowest to the highest expression of CPA1. And by looking at the plot, we can see a general positive correlation between CPA1 and PSSR2 expression. And because it's colored by disease state, we can see each of the colors of the disease states on this plot. And by highlighting over each of the disease states, we can see that for it doesn't matter which disease state the donor is in, the positive correlation between CPA1 and PSSR2 is still there. So next, let's say that we're only interested in control alpha and beta cells. What we're able to do is select just alpha and beta, and then control. Press this slice and apply button, and now we subsetted the data so that we only see the alpha and beta control cells. I'm going to use this magnifying glass here to pan over and zoom into the data so we can see more detail. Zoom out more. Okay. And now I'm going to run differential expression again between the alpha and beta cells. But this time I'm going to use the lasso tool. So I'm going to select this icon here, and then draw a circle around the left side, which is the alpha cells, and have that assign that as the first group. And then I'm going to draw a circle around the right side and assign that as the second group. I'm going to press the Venn diagram button, and now the differential expression is going to run again. So one thing that we have a lot of different gene sets at this point because of all the differential expression. So I'm going to click this triple dot icon here, edit gene set name. I'm going to call this alpha or control alpha. I'm going to call this control beta. I'm going to call this control SNR. Okay. And I'm going to confirm that the differential expression between alpha and beta cells was correct, because the top gene for alpha cells is GCG, which is glucagon, and then the top gene for beta cells is insulin.